Dear all, this is Martin Hofmann-Apitius, recording my talk for the EN eBrains Joint Workshop, The Future of Medical Data Sharing in Clinical Neurosciences. My name is presented here again. I'm with the Fraunhofer Institute for Algorithms and Scientific Computing in St. Augustin, close to Bonn in Germany, and I'll be talking about ontologies for clinical neurosciences. We cover in our department a wide span of technologies that actually assemble into what we call the value chain. We are quite involved in unstructured information mining, knowledge graph technologies. We focus also on making knowledge graph and making formalized knowledge reusable in models combining data and knowledge. And ultimately, we focus on using AI for actionable insights and decision support. So quite a wide span. Coming back to ontologies for clinical neurosciences, probably makes sense to briefly recapitulate what ontologies are and what they are good for. Ontologies are formal representations of terms, concepts, and their relationships. And the cool thing with ontologies is actually that they can be read and understood by humans, and they can be read and used by machines. And therefore we find ontology when it comes to, for instance, annotating data, so-called metadata annotation. They are also quite helpful when we try to integrate different types of data using ontologies as what's called semantic glue, typically an abstraction layer represented or implemented by ontologies. Ontologies can be used as templates for approaches in text mining, unstructured information mining, and they are also quite good, and we know them actually predominantly from the enrichment analysis that are done typically in gene expression analysis, GO-term enrichment and so on. You have heard that before. So we all witnessed the major impact that one particular ontology, the gene ontology, has on all the functional interpretation of patterns in omics data in particular. Now, my team at SCAI has been working on ontologies for many, many years, including common ontologies. What you see here is an example of the clinical trial ontology, the CTO, that we have recently updated and extended with the help of the FDA, colleagues at the FDA, the NCBI, and the University of Michigan. That is a formal representation of everything that is important for clinical trials, and it was made with the idea in mind to link the world of PubChem to the world of clinicaltrials.gov at the NCBI. In the neuroscience field, we are a bit more, better represented in that world with a lot of ontologies. Most recently, the Epilepsy ontology, which is actually integrated a couple of pre-existing ontologies that were rather incomplete or that obviously benefit from the integration that we did. We did that together with major stakeholders in the epilepsy field, including the International League Against Epilepsy. And that is now being released very soon publicly and can be used for a standardized and controlled vocabulary in the epilepsy clinical research field. I don't have really the time to walk you through the entire upper level structure of the epilepsy ontology that's shown here, but the most important points are that both the world of research and biomarkers and the world of clinical practice are both represented in one ontology, and it links to major widely used and adopted standards in the medical informatics field, which is quite important because we want to use that also to bridge into the clinical routine data, the EHR world, for instance. We in our team at SCAI use ontologies frequently for information extraction. This is just one example where you see the highlighting the markup of ontology terms in scientific text, and you can use that for retrieval and information extraction. Another nice example for the use of an ontology and the tailored development of an ontology comes from our colleagues at the Demenzias platform UK, DPUK. This is a slide that Sarah Bauermeister was kindly providing to me. It shows how the data sets, these are dementia data sets, these are neurodegenerative disease data sets, in the DPUK are characterized by a set of terms and categories that are assembled in the so-called C-SURF ontology. And they established even an encoding system based on ontology terms and on hierarchical levels that depict or that encode essentially the path to a specific object, a specific variable in a given data set. This is a way to enable ultimately identifying related data sets in the DPUK. We did a very similar approach down to the variable level in the field of Alzheimer, so highly related. That's why we collaborate with DPUK. We implemented the probably most comprehensive overview on widely used longitudinal Alzheimer study data. This is actually work done in the context of the TVB Cloud project that Petra Ritter is leading. And in this case here, we have actually mapped at variable level all the major longitudinal AD data sets worldwide. These are 20 data sets onto each other. And that was an immense effort on the side of data understanding, data curation, data mapping. But the tool is freely available and it's purely metadata. So you can have access to a lot of information about the data in the studies, very detailed information, without having the real patient level data at hand. And we are not allowed to share the patient level data, but we provide you with the maximum of information on the studies, and they are fully comparable at individual variable level. Now, if we want to manage and to use ontologies, we need to handle them in a way that they can be managed, versioned, for instance, that they can be aligned, that they can be chosen and found, make them usable. And that's what we did in a small project in the context of the Human Brain Project. There we implemented a referential ontology hub, which is actually building on a framework developed by the colleagues at the European Bioinformatics Institute. Helen Parkinson's team has created the ontology lookup service. This is the ontology hub of the EBI, and we reused that technology, and thanks Helen for sharing that with the rest of the world. And we reused that and implemented it in a dedicated version for ontologies relevant for computational neurosciences. On our side, on our SCAI side internally, we have even further integrated services that utilize ontologies. First of all, we have taken care of all the versioning. Here in the core, in the center, is the EBI's OLS engine that can import public ontologies from BioPortal or the OBO Foundry, for instance. So for repositories of ontologies, we do proper versioning. We know when we import it and what version that is. We have a mapping engine that links out from the ontology terms to major standards in medicine, for instance, and also maps to entries in important biodatabases as far as that is possible. And the entire mapping engine linked to the OLS is used by our information extraction engines, in particular the text mining engine SkyView. And that in turn actually is now used by Petra Ritter and her team to populate a 3D model of the human brain, the virtual brain, with information coming from textual sources. So there we have a direct link between semantics on one side, text mining on the other side, and a 3D representation and an entire simulation engine for the human brain on the other side. That's an absolutely cool development they are doing in Berlin. This is just an overview on some of our work during the recent years. You see that we have a long-standing agenda in creating various different ontologies, formalized representations of knowledge. When the pandemic struck us, we even created a COVID-19 ontology in less than 3 months. And currently there's a multilingual version underway. But we have been focusing on neurodegeneration, and that shows by the Alzheimer, the Parkinson, the multiple sclerosis, epilepsy ontology. We extended that towards psychiatry now with a schizophrenia and PTSD and traumatic brain injury ontology. What can we use it for? What are we doing with that? Well, I mean, in the end, we want to use ontologies. It is fun to create them, but ontologies are not good or bad. They are used or not used. And I show you examples how we use ontologies now. This is just an example how you can use ontologies in a text mining environment to retrieve specific drugs that are mentioned in a certain context. That's not so super cool. That's standard in text mining. But what's really cool is using this retrieval and information extraction to actually represent knowledge in a new way. And this new way is the knowledge graph, the computable knowledge graph, that turns a sentence like this one here, phosphorylation of glycogen, synthase kinase 3 beta at threonine 668 increases the degradation of amyloid precursor protein into a formalism like this one here. This is what we call a triple. It is one object, it has a relationship here, and it's another object. We have some modifiers, some descriptors that are specifying what happens to these objects, but essentially this is a triple. Yeah? And triples form graphs. We have actually generated gigantic graphs. We are now generating graphs that cover entire indication areas. Everything that's published on epilepsy, everything that's published on Alzheimer's. And here's an example. Started that work in 2014, 2015, and this is from Alpha Tom Kodamolil's groundbreaking work on a computable model for Alzheimer, which in the end now in its final state contains 124 subgraphs representing 124 Alzheimer disease mechanisms. And the cool thing is they are multi-scale. They are inherently going across multiple scales. They are not pathways. Pathways are typically monomodal, and they don't extend like from the molecular level to the biological process level and even the clinical level, but our graphs actually do that. Our graphs span from genetic variation to imaging readouts or cognition readouts. And this is what you can do with a computable knowledge graph. We have used that in various different applications, these knowledge graphs, including stratification of patient-level data based on mechanisms, so mechanism-based stratification, which was the subject and the core goal of the Autonomy project. But we didn't stop there. So what we have been doing now recently was an enrichment of our knowledge graphs with additional highly curated information, pathways, but also entries from databases. And we enriched the entire knowledge graph with drug target information in an integrative framework, which is the EBEL framework developed by Christian Ebeling. And what results from this gigantic graph, full of cause and effect statements, enriched and populated with chemistry, bioactive chemistry, is what we call a Pharmacome. The human brain Pharmacome currently is full of pathophysiology mechanisms and drug target information linked to these pathophysiology mechanisms. And the work that we did there is massively empowered or enabled by ontologies and terminologies underlying, so clear and formalized semantics, leading to a huge computable graph, which looks like this and has quite impressive statistics if you look into the number of genes, more than 11,000 genes in it, more than 10,000 proteins in it, a lot of relationships, pathology associations, cell type associations, cellular localization expressions, and so on. A variety of impressive expressiveness when it comes to the types of relationships. So that is a gigantic functional network of the human brain, and it's the bioactive substances active in the human brain. And that's the human brain pharmacome. We were interested in a particular aspect of the human brain pharmacome, which is the identification of drugs regulating the post-translational modification of pTAU. Tau we all know from you know, if you're in neuroscience, you know, of course, tau. This is the tau subgraph that we identified, that we extracted from the human brain pharmacome, from this giant galaxy in space, and it's still quite impressive. This is the pTAU mechanisms plus the drugs, so populated and highlighted for the drugs that somehow potentially influence the post-translational modification of tau. Now our aim was to find drugs that we could repurpose that influence on the post-translational modification of tau for, I think, obvious reasons. We want to identify easy wins on the pharmacology side to play with the tau species in the brain, essentially. So the workflow for the identification of secondary targets, pTAU modification modifiers of pTAU post-translational modification, was done as follows. We extracted, as I showed you, all the phosphorylated species of the tau protein from the knowledge base. We identified everything that is somehow a upstream regulator, so proteins linked in a causal relationship to tau phosphorylation. These were 82 proteins. Amongst those are 43 druggable proteins that have either ligands, known ligands, or have properties that make them, render them essentially druggable proteins and qualify them. And then we use that core graph essentially to enrich it again a little with dedicated literature searches, and added a little bit of additional information, interesting information. And why we did this becomes obvious here in the next slide, where we show the primary target, which is the pTAU, and we have a couple of secondary targets that either qualify directly or not to become targets for modification of post-translational modification. In some cases, the upstream regulatory link was unclear, and through the literature mining, the additional literature mining, we could actually reconstruct the path that leads from such a target to the tau target and the post-translational modification mechanisms around tau. To cut the story short, we have identified HDAC6 as one of the key modulators and the most interesting modulators of pTAU post-translational modification. And we have set up together with our friends and colleagues at the Fraunhofer ITMP in Hamburg. This is the European Screening Port folks, Carsten Clausen and friends. We have set up a assay that discriminates between one HDAC6 activity, which is histone-specific activities, and the tau-specific activities, and selected drug repurposing candidates that are excellent modulators, should be excellent modulators of pTAU post-translational modification and phosphorylation. And that's currently in revision, that paper describing the entire Pharmacome approach and the drug screening. And we have more than 20 interesting confirmed hits in the repurposing screen. And that's quite encouraging. That's where we stand with making use of a semantic graph, if you want. Now, what's the take-home message? What I want you to take home from my talk is that ontologies are widely known for their use in various types of enrichment analysis, but I hope I could convince you that the application spectrum for ontologies is much wider. Ontologies are useful for data and knowledge harmonization. They can be used for information extraction and text mining. They serve in our knowledge graphs as semantic invariables that we can refer to. They are helpful when it comes to making data and knowledge widely interoperable, almost indistinguishable anymore. And I've given you a rather concrete example how shared and formal semantics can go way beyond enrichment analysis. They are actually essential elements of knowledge graphs and form the basis for entire pharmacomes. And with that, I'm happy to take questions and thank you for your attention.